Today we are going to learn how to extract DNA from a strawberry. DNA, or deoxyribonucleic acid, is a special molecule that provides instructions for life as we know it. A DNA molecule has a very characteristic double helix structure. Nucleotides, or bases, comprise the center of the DNA molecule and are held together by what's called a backbone. Specific combinations or sequences of nucleotides or bases are called genes. Genes are what determine things like eye color, height, or even whether we can roll our tongues. In a cell, like this strawberry cell shown here, DNA can be found inside a structure called the nucleus. The nucleus and other cellular structures are protected by something called the cell wall. The cell wall is a rigid structure that helps the cell keep its shape. Today, we are going to extract DNA from the nucleus, which lives behind this protective cell wall. To extract the DNA from a strawberry cell, we first need to make a solution called an extraction solution. An extraction solution consists of water, soap, and salt. When we add our strawberries to this extraction solution, let's see what happens. The soap helps to break down the rigid and protective cell wall, as well as any membranes surrounding the nucleus. You can think of the soap as if it's creating doorways in the cell wall and in the nucleus. These doorways allow pathways for the DNA to flow out of the nucleus and into the extraction solution. The salt in our extraction solution allows the DNA that has flowed out of the nucleus to clump together. This will allow us to visualize the microscopic DNA. Finally, we'll add isopropyl alcohol, also known as rubbing alcohol, to our extraction solution. When added to the extraction solution, the isopropyl alcohol will not mix with the water, much the same way oil and water don't mix. The DNA also cannot mix with the isopropyl alcohol, and as a result, both the DNA molecules and the isopropyl alcohol will float to the top of the container, while the extraction liquid containing the water and the strawberry cells will stay at the bottom. The DNA will form a white stringy mixture, also called precipitate, which we can then extract from the top of the isopropyl layer. Now that we know how DNA extraction works, let's try it out ourselves. For this activity, you're going to need a source of DNA. Strawberries, either fresh or frozen, work well here. You'll also need some water, salt, and dish soap for your extraction solution, some isopropyl alcohol or rubbing alcohol, as well as some small containers for mixing and extracting your DNA, a plastic Ziploc bag, a strainer or paper towels for straining if you don't have one, a small stick, and a spoon for mixing. To get started, you'll want to put your isopropyl or rubbing alcohol in the freezer. You'll want your isopropyl to be ice cold before adding it to your extraction solution. To make your extraction solution, mix 1/3 of a cup of water with 1 tablespoon of dish soap and half a teaspoon of salt. Mix well to combine. Next, we'll want to add our strawberries to a plastic bag. We're going to mush these strawberries up well before adding our extraction liquid to help us extract as much DNA as possible. Before mushing up your strawberries, make sure that all of the air has been squeezed out of your bag and it is sealed tight. Mush the strawberries in your bag until there are no whole pieces left and you have a liquid consistency. Next, add about 1/3 of your extraction solution to your plastic bag. Seal and mix well to combine. Remember that our extraction solution works by creating doorways in our cell wall and nuclear membranes. The DNA can then move from the nucleus into our extraction solution. We will isolate the DNA in the extraction solution by straining out the larger pieces of strawberry cells that remain. If If you don't have a strainer, you can strain this mixture through a paper towel. Just be aware that this process could take some time. Once you have just your extraction solution containing your DNA, it's time to add the isopropyl or rubbing alcohol. Add a small amount of isopropyl alcohol to your container containing your extraction solution and DNA. Carefully pour in the isopropyl alcohol by pouring along the side of the wall of your container. Allow to sit for a couple of minutes or until you begin to see a white, fluffy, or stringy material forming at the top of your container. As you can see here, when I swirl just the top layer of my solution with the skewer, the white material is collecting. This is your strawberry DNA. Remember that your isopropyl alcohol and DNA form a separate layer at the top of your extraction solution. Therefore, as you're swirling around your DNA in your container, make sure that you're not going too deep. The DNA will be sitting on the surface of your solution. There you have it. That's how you extract DNA from a strawberry.